Okay. We are recording now, so everybody's going to hear your background chatter, I hope. The difficulty with the lectures arose because they had done some renovations or expansions of the sound system in here, and when they did that, they didn't completely reconnect everything quite the way it was before, or there are new things in the loop, or something along that line. I have been assured by 2 different people of MedAV that we are now functional again, so hopefully what we hear and say today will be a video of the old style at the end of the lecture. For the lectures intervening, which did not have the usual capture, I have posted links to last year's lectures that were on that subject on Google. Be aware that they have some references to course material and stuff if that is not relevant to what we're doing this year, so skip over it. I have also posted the PowerPoint from Wednesday when we were relocated because that auditorium did not have a recording device like the one we have here. I'm going to tweak that down just a little. Is that still pretty clear there in the back? Yes? Good? Okay. Do we have any questions about nitrogen metabolism before we go ahead with the other topics?
Do you want to ask people what day they'd like the review session?
All right. We're looking ahead to a review session next week before the 3rd hour exam. The 3rd hour exam will be a week from today here. And were Wednesday and Thursday the choices, Tim? Yeah. And we're prepared to offer either on a Wednesday or on a Thursday. So I'm going to ask you if you'd be willing to come, if you're interested and willing to come on a Wednesday, please hold up your hands. Hi. Tim, take a look. If you're interested and willing and you'd come on a Thursday, please hold up your hands. So that looks like there's a bit more usefulness to a Wednesday date, to my eye. There will also, of course, be office hours between me and Lynn and Tim. So there's lots of opportunities to get your questions answered if you can't make a Wednesday session. And we will announce the exact time and place of that Wednesday session very shortly. Any other practical questions? It'll cover, as the syllabus said, it will be on chapters 11 through 15, citric acid cycle, electron transport, photosynthesis, lipids, nitrogen. Any other questions about the exam before then? Yes?
Will you be posting the practice exam and an exam summary?
We'll probably be posting a practice exam by the exam summary. Do you mean was that essentially the same as the handout for the review session, I think? Yeah. I imagine we'll have one available by the time of the review session. Whether we'll have one available earlier, I don't know. Usually Brenda would post it today, so we'll see. Other questions? Okay. So we closed up our discussions of amino acid metabolism or synthesis with histidine, which is a peculiar amino acid in that it borrows aspects of nucleotide metabolism. And so it's a nice kind of crossover point to move ahead to some other aspects of nitrogen metabolism. Now, we're used to thinking of amino acids as being the building blocks for proteins, and they certainly are. But amino acids are also the starting material for many of the neurotransmitters. In some cases, the amino acids themselves are neurotransmitters. So glycine, glutamate, and aspartate all have receptors on neurons that lead to electrical excitation. And in fact, an excess of glutamate can actually be toxic to nerves. You might think, why would you use amino acids as a neurotransmitter? Some of these receptors are present on single-celled organisms and were probably used by them to find locations that had a lot of amino acids to have a happy life. And so this is an example of using a device for a new purpose later on. Some of the other amino acids are arrived at by slightly more indirect routes. So for example, Glutamate can be decarboxylated, that is, you can shed a carbon dioxide and make GABA, which is short for gamma-aminobutyrate, methane, ethane, propane, butane, and it's alpha, beta, gamma, so it's gamma-aminobutyrate, or GABA. GABA is a major inhibitory neurotransmitter, and it plays an important role in anxiety diseases. Many of the anti-anxiety drugs, in fact, involve GABA metabolism. Some of the other amino acids that are involved are also important. So tyrosine, here we see tyrosine, standard amino acid, as if it were an alanine, phenyl ring, and an alcohol. And you can see some similarities between tyrosine itself and the compound dopamine. Dopamine has an extra alcohol group. It's cast off that carboxylic acid, but its family resemblance is strong. Dopamine is the neurotransmitter involved in Parkinson's. Deficiencies cause Parkinson's. A wonderful, wonderful book called Awakenings by Oliver Sacks, discussing the clinical impact of that discovery that you could overcome Parkinson's with the precursor L-DOPA. This is also one of the diseases in which cell therapies seem to be have a potential role, that Parkinson's in mice can be overcome with brain transplants of dopamine-producing neurons. Tyrosine is not the only, it does not just go to dopamine. It is further processed into two other transmitters, norepinephrine and epinephrine, or if you may recognize them as noradrenaline and adrenaline. These are produced in the adrenal glands and the kidneys. That's where they get their names from. But they're present in a wide variety of body tissues. They play a role in recruiting glucose in the muscles. They also conspicuously seem to be involved in depression, or may be involved in depression. And these are collectively called the catecholamines. You can see that this is catechol, and here these are catecholamines. Another amino acid with an important role is tryptophan. And this is just reminding you about the structure of tryptophan. And with a decarboxylation, like we saw in making dopamine, and then hydroxylation, we now have serotonin. And you may recognize serotonin. Serotonin is another neurotransmitter which is believed to have some kind of a role in depression. The somewhat notorious antidepressant Prozac is a reuptake inhibitor. You remember we talked back on neurotransmission about bridging the synaptic gap. Drugs like Prozac prevent that reuptake, so what you have stays around longer. It's worth mentioning at this point that although the ability of Prozac to prolong serotonin reuptake is well established, its mechanism in reducing depression is not particularly well understood because the antidepressant effects take several weeks to take effect. So if it was simply a question of how much serotonin there was in the neural gaps, that would happen in a matter of hours. Serotonin is itself a precursor for yet another transmitter called melatonin. You can again see sort of the backbone here of the serotonin that's been modified lightly. And melatonin is involved in circadian rhythms and in the level of pigmentation in the body. So amino acids are really flexible, diverse kind of compounds when it comes to neurotransmitters. Yet another amino acid that can contribute these communication molecules is arginine. And here you see your arginine molecule again. The terminal group is sometimes called a guanidino group. And it's going to produce nitric oxide, NO. And to get to that point, we were going to have to do a certain amount of oxidation. And so you're coming in with NADPH and an oxygen molecule. You now have a single oxygen bonded to that nitrogen. You come in again with NADPH, and ultimately we have left behind a citrulline molecule in which we've liberated an NO radical. Nitric oxide is a neurotransmitter. It can talk from one nerve to another. It's also much more widely active in the body. It can act directly without the presence of nerves on muscles, on the heart, on the immune system. The pain called angina pectoris, which is a consequence of spasms in the heart arteries, that has been treated literally for decades, over a century, with nitroglycerin. And there's the structure of nitroglycerin. It was observed very early on that very small amounts of nitroglycerin would relieve this pain, and it was only quite recently that we found out that it's the nitric oxide that accounts for that. So we've talked a bit there about amino acids as having a very distinctive separate role as informational molecules on a very local scale. The other large group of nitrogen-containing compounds are the nucleotides. And nucleotides are something you can't do without. And you can't depend on getting very much of them in your diet. There aren't very many foods that have more than a small amount of nucleotides in them. And so the body has not taken the strategy that it did with amino acids. It has preserved pathways for making all of the nucleotides. In fact, it's gone sort of to a built-in suspenders approach. It not only can synthesize them, but it can also salvage them when they're done with the older forms of it. We can take in nucleotides from the diet. Typically speaking, foods that are rich in nucleotides are ones that are very new forms of life. So mushrooms, broccoli, these are things that have a, relatively speaking, higher proportion of nucleotides. So there are a couple of strategies. The purines take one particular one. That would include AMP and GMP, adenosine monophosphate, guanosine monophosphate. They both build on a 5-phosphoribosyl pyrophosphate base, PRPP, since it's so hard to say. And we mentioned PRPP already when we were talking about the synthesis of histidine. And so the first thing we have to make is PRPP. It starts from ribose 5-phosphate, so this would be a product of the pentose phosphate pathway. It consumes an ATP down to the level of AMP, so it takes 2 high-energy bonds to make the 5-ribosyl pyrophosphate, the PRPP. So you've got phosphates hanging off both ends there of your ring. And the enzyme is rational, ribose phosphate pyrophosphokinase. Purine synthesis, since we're making 2 fused rings that are heterocycles in both of them, as you can imagine, takes a number of steps. And we are not going to go through every step of those 10 steps in the pathway. In addition, to make purines, you need at least 5 different molecules contributing building bits to it. And in the breakdown of that goes something like this. I suspect that the Xerox copies are not quite as colorful. But you can double check this in the textbook if you like later on. We see some components coming in from amino acids. So aspartate contributes a nitrogen. Glycine contributes a nitrogen and carbon. Glutamine is contributing a couple of nitrogens. We get in a bicarbonate coming in, and a couple that are coming from tetrahydrofolate. So here's formal tetrahydrofolate there and there. So we're getting contributions from several different directions, but the amino acids are still very important in it. And this is not either of them yet. This is inosine monophosphate. Inosine is kind of like a neutral precursor. We can make either one out of the inosine. But we have now our purine ring. Now, there's a very cute trick here. We have our inosine monophosphate. So here's the inosine ring from inosine monophosphate. And we want to ultimately make both AMP, adenosine monophosphate, and GMP, guanosine monophosphate. They're they're structurally fairly similar. There's an additional nitrogen on each of them, additional oxygen on the guanine. So there's not a lot to be done, but there's a bit. And the cute trick is that we're going to see that each compound ends up controlling the opposite pathway. In general, you're going to want as much ATP as GTP and so on in terms of nucleic acid synthesis. And so you want to be able to keep the production of them balanced. And let's see how that works. Here we have our IMP, inosine monophosphate. In the next step, we're going to use aspartate to contribute its whole self there, spending a GTP in the process. This is adenylyl succinate. By splitting off the carbon backbone, that part of it, we deposit a nitrogen there on the ring, leaving off a fumarate, and now we have AMP. So that's a nice trick. But you notice we use GTP to make the adenylyl succinate. On the other fork of the pathway, we're going to do a little bit of oxidizing, so we're going to use NAD plus here to start the oxidation process. And here we have xanthosine monophosphate. In this case, glutamine is going to contribute nitrogens in this case, and that reaction will be paid for through ATP, going down all the way to AMP and inorganic phosphate, which is then split. And then the product of that is GMP. So very cutely, having each of the nucleotides necessary for the opposite one to be made helps keep them in balance. And since both aspartate and glutamine are contributing the nitrogens, it helps to couple the rate of purine synthesis to the availability of amino acids for biosynthesis. So you have sort of built into it a certain amount of sensitivity to the overall status of the organism. Pyrimidines have their own approach. They look simpler, certainly, to make, so there's probably going to be less that goes into it. One of the things that's a different approach to it here is instead of starting from a PRPP base and then slowly building up the pieces on it, like we did for the purines, in the case of the pyrimidines, we're going to make the base and then slap it onto a PRPP later on. And where do we get the bits and pieces for the pyrimidine ring? We have, again, glutamine playing a role. Aspartate, in this case, is sacrificing its whole self, basically, here, right? And we have a carbon coming in from a bicarbonate molecule as well. And so once that has been synthesized, it can be put onto the PRPP base, and here what we see is uridine monophosphate. So the synthesis of pyrimidines is sensitive to the presence of any of the uridine nucleotides, UMP, UDP, UTP, but it would be activated by ATP. So if you're having an abundance of ATP, maybe you're falling behind on your pyrimidines and it's time to up your synthesis there. CTP is made from UTP. So here you have your UTP. We just made that base and slapped it onto the PRPP. And glutamine is going to contribute a nitrogen. It's going to cost us the ATP, and now we have CTP here with the nitrogen where we had an oxygen before. Now, what we've been making so far are all ribonucleotides. They have ribose as their sugar moiety. And ribonucleotides are fine if what you want to make is ribonucleic acids like messenger RNA or transfer RNA or ribosomal RNA. But for DNA synthesis, we're going to need the deoxynucleotides. And to do that, we're going to have to somehow or other make that 2' position lose its alcohol group. And that's accomplished by an enzyme called ribonucleotide reductase. That this is a mildly oxidized carbon here. To get it to the state we need for the DNA, it has to be down to a hydrogen there. And the enzymatic reactions involved in this are fairly slick. It's going to exploit a number of intermediates that are a little bit almost flashy, if you like. There's going to be stages in the process where you actually have a radical on the ribose ring rather than an ion or something like that. The enzyme starts off with a free cysteine radical. The dot there on the S is a radical. And it can work by abstracting the hydrogen only, leaving this as a radical. And then in the next stage, the middle of the sulfhydryls is going to donate a proton at this position, and now we have a positively charged moiety there, and it becomes the radical here is helping to make this, what they call carbocation, somewhat more stable, and it's free to leave. This, when it departs, comes off just as water, leaving the cation there stabilized by the radical. And by yet another proton and electron coming off of the enzyme itself, we can restore this to an H and an H. We still have a radical over there, so we probably want to do something about it. And in the final stages, what we accomplish is by abstracting that proton back, we can restore it, and the enzyme is back into its original state, except for the disulfide there, which we can use thioredoxin to reduce down. And so this goes in a cycle and can keep doing it as often as possible. We're also going to need thymidine if we want to make DNA. And thymidylate synthase is an enzyme that can take deoxy-UMP. Now, deoxy-UMP would not normally be used for anything. It's not right for RNA. It's not right for DNA. And thymidylate synthase will take the deoxy-UMP and add a methyl group to it. So here's your deoxy-UMP, or dump. And I know, some of them have easier to read names than others. Here's the tetrahydrofolate with, in this case, a methylene L reduction state. And it's going to contribute that as a methyl group right there. So the tetrahydrofolate is going to add the methyl group. Once that's happened, that's going to have to be reduced to regenerate that. And so things that interfere with that are going to interfere with the production of deoxythymidine monophosphate. If you don't have deoxythymidine monophosphate, you're not going to make DNA. So inhibitors of this reaction are anti-proliferative, and a common one that is used in that way is methotrexate. Many of you may have heard of methotrexate. It's a very broad spectrum anti-proliferative drug for this reason, that it prevents the formation of the DTMP that's needed for DNA synthesis. And those reactions are again on a bifunctional enzyme, which is kind of fun, and are to move the dihydrofolate along the surface of it. It has an electrostatic highway that allows the cofactor to be moved up to the second functional activity. Okay. So far we've been talking about synthesis. Obviously, in order to make ourselves, we have to be able to make the amino acids or get them. But there are times when we have an excess of amino acids, if our diet is particularly rich in nitrogen, in protein. There are also times such as fasting, when we need to use our own muscles as a source of energy. Interestingly, the small intestine has as its principal fuel amino acids. So although we tend to think of glucose as the fuel, in fact, different tissues in the body have a surprising amount of variety of what they like. And in the case of the small intestine, it's amino acids. The amino acids that make it past the small intestine are going to be processed first at the liver, And the liver is this wonderfully integrative metabolic organ. It may use them directly as fuel. It may use the carbon backbones from the amino acids in gluconeogenesis and make more glucose. It can use the carbon backbones to make ketone bodies. In fasting, the muscle breakdown that provides the energy starts with the skeletal muscles. If the fasts are too prolonged or too extreme, it starts attacking the heart muscle. And one of the causes of death from starvation is, in fact, heart failure. We're going to see that amino acids can be used to generate either sugar or fat. And for that reason, we call them glucogenic, ketogenic, or both. So some of them, by the structure of the backbone, can end up back in glucose. Others can only end up in ketone bodies or fatty acids. So let's look at how we take some of these things down. If we start with asparagine, the first step in its degradation, if we want to use this for a fuel or to get rid of it, is asparaginase, which you can see releases an ammonia, an ammonium ion. So this is letting go of something we put a lot of energy into getting. So obviously burning fatty acids is not necessarily the first thing you choose to do. By taking off the amide nitrogen, you've now made aspartate. Aspartate is then the substrate for transaminase, and alpha-ketoglutarate can come in to receive that nitrogen and release a carbon backbone of oxaloacetate at that point. Glutamine, just one carbon longer, can also lose its nitrogen through glutaminase, giving you glutamate. And glutamate can transfer its amino acids also, but in that case you're not ending up with any net because you still have a glutamate, and an alpha-ketoglutarate. This does mean that a glutamate ends up as kind of a common endpoint for nitrogens you want to get rid of, and we're going to see what happens to that glutamate in a little bit. Serine and cysteine we saw came from pyruvate, and they are each of them 3-carbon backbones. And so we're not too surprised to find that they end up producing a 3-carbon backbone, specifically pyruvate. And so if we look at serine, it's extremely easy to get from serine to pyruvate. Just simply the loss of the ammonia gives you pyruvate. I flipped this image so we could fit all of it onto a single slide. Here's your cysteine. When it loses, it'll have to lose its nitrogen as an ammonium. It also is going to need to lose that sulfur as a hydrogen sulfide. And we're not going to go into the metabolism of sulfur excretion. Although there is metabolism of it, I'm just not going to go into it. So this is to give you an overview of the variety of amino acids that potentially end up in glucose. Whether they actually end up in glucose depends on the whole metabolic status of the organism, and we'll be talking in Chapter 16 about that integration of metabolism. But these are the ones that have the potential to end up in glucose. We just talked about serine and cysteine as 3-carbon amino acids that go to pyruvate. We saw earlier pyruvate carboxylase can generate the 4-carbon oxaloacetate. And we know from our earlier studies that oxaloacetate can be used for gluconeogenesis. Aspartate and asparagine are already 4 carbons. They go directly to oxaloacetate. The 5-carbon amino acids, glutamate and glutamine, enter through alpha-ketoglutarate, and the citric acid cycle does the work of taking that down to oxaloacetate, doing some other useful things along the way. So we can see here that we have 1, 2, 3, 4, 5. We have 6 amino acids, all of which can end up at glucose. There also are all amino acids with a fairly simple structure to them. Threonine is kind of an interesting, tricky little thing. Here's the structure of threonine. Remember, it's you could think of it as serine with a methyl group added is one way of thinking of threonine. And we start by oxidizing this a bit. So we take that alcohol down to a carbonyl oxygen, producing a little bit of NADH, which we could use for energy, of course. And now what we have is alpha amino beta ketobutyrate, so it's a 4-carbon backbone. And with a CoA coming in, this is set up in a way that it allows the formation of a molecule of acetyl-CoA, and what's left is simply a molecule of glycine. So the glycine can go off and be used the way the way the glycine can to make pyruvate, and the acetyl-CoA can go off and get used, as we saw earlier, that acetyl-CoA can get used to make the ketone bodies. We mentioned that glycine can be made into serine and then into pyruvate, but the principal way in which glycine is degraded is somewhat different. So here we have the glycine molecule, the simplest of all the amino acids, and there's a glycine cleavage system that uses glycine as a source for making methylene tetrahydrofolate. And then what's left after that is ammonia and a carbon dioxide. So the methylene tetrahydrofolate is quite useful. The ammonia we're going to deal with in a little bit when we get into how ammonia gets removed from the body. Valine is a little trickier. So here you see our valine molecule. It's the amino acid here with a V-shaped side chain. Alpha ketoglutarate comes in to receive its ammonia, coming off as glutamate. So it's again serving as the receiving thing for the nitrogen. And what's left is the branch chain alpha keto acid. So it's an alpha keto acid with a branch chain. It's not exactly quite as nice as the things we've been playing with in the citric acid cycle because it has this branched chain in there. But it is, in fact, can be treated by a dehydrogenase complex that is the branch chain dehydrogenase. Now, we talked about pyruvate dehydrogenase, and we talked about alpha ketoglutarate dehydrogenase and how those two have rather similar structures and ways of doing it. The branch chain alpha keto acid dehydrogenase does a very similar kind of reaction in which it also produces a CoA compound, now with a branch chain, casting off a carbon dioxide and an NADH. But it's still a branch chain. We've got to do something with it. And we have we can further reduce that to make a reduced quinone, and you see the double bond in there now, and then that can then be turned into succinyl-CoA. And succinyl-CoA, of course, is one of the intermediates in the citric acid cycle, a potential precursor to the heme molecule or a potential precursor for gluconeogenesis. It's interesting. They're describing it as ketogenic, and yet it's going into succinyl-CoA. So I think you might be right about that. That looks like it probably is a typo here. Sorry, what? So in other words, I think what he's pointing out is this probably should read glucogenic if we're producing succinyl-CoA. This is the metabolic error that leads to a disease called maple syrup urine deficiency disease, which was first identified by a Norwegian mother who had 2 babies whose diapers smelled like maple syrup. It's like, what? And she was very persistent, and she kept after the biochemists and the pediatricians till they figured out what the basis of the disease was. I'm trying to think if I can correct that here or not. Let me just write a note to that effect, and I'll deal with it later. This is 39, huh? Is there other questions? So we'll pursue that. Are there other questions? Okay. Now, some of the amino acids look a lot fancier than alanine or serine or whatever, especially the aromatic amino acids. Just as we didn't go into all the details of aromatic amino acid synthesis, we're not going to go through all the details of their degradation, but there are some important aspects to it. First of all, the aromatic amino acids of phenylalanine and tyrosine are both glucogenic and ketogenic. They're going to end up producing acetoacetate, which we saw before in ketone synthesis, and either alanine or fumarate. It looks something like this. You've got phenylalanine, which needs to be, which we can turn into tyrosine with phenylalanine hydroxylase. Once you have the tyrosine, a transaminase turns that amine group into a carbonyl oxygen. And then a dioxygenase molecule can add oxygen across this ring. So you see you had one alcohol group here, and now you've got two oxygens there, and you've lost the carbon dioxide up there, helping to drive it. And then this molecule can be opened up into a linear molecule, this molecule itself is called homogentistate, and it can be opened up into the full mallyl acetyl. Now, this enzyme and this enzyme were also among some of the early inborn errors of metabolism. And they were discovered by excess in the urine of some of these compounds that couldn't be degraded along the way. They are fatal if not treated. They are treated dietarily, where you make sure that the infants get the absolute minimum of aromatic amino acids so that they don't have to degrade any more than they have to. Why aren't they lethal to the fetus, before the fetus is born? Sorry, I think I heard it. What?
The mother's already degraded them?
The mother takes care of a lot of this work for the fetus. So the mother's kidneys really handle a lot of the work of, and liver, handle a lot of the work of detoxifying things during pregnancy. Well, we've been making ammonia after ammonia after ammonia in this degradation process, and we haven't done anything with it yet. We can't just keep the ammonia ions around. A little bit of ammonia can be used by the kidney to excrete acid compounds in an electrically neutral way. But it can't be kept for very long, and it can't be kept in very high levels. Ultimately, ammonia is toxic. It will change the blood pH to an alkaline pH. We're going to see how it can influence the citric acid cycle itself. So we've got to do something with the nitrogen. And what we do with the nitrogen, what most mammals do with the nitrogen, is make urea. And here's the structure of urea, very simple compound. This was the first organic molecule that was completely synthesized from inorganic compounds. The liver does most of this work. Kidney can do some of it. And urea has a number of properties that are very nice. It's extremely water soluble. It's dense in nitrogen, so basically half of the molecular weight of it is nitrogen, pretty close. And it's not very interactive. It doesn't do much of anything else. So how do we get there? Okay. We've been taking glutamate molecules and oxyl acetate molecules and using them to pick up nitrogen and making glutamate. So we are generating this kind of pool of glutamate that was our dumping ground for nitrogens. To get the ammonia off of that glutamate, and we have enzymes in the mitochondrion that can release that. They do so at the expense of either NADP or NADPH. So this is unusual. This is the only enzyme that is known to use either or in this case. And what it ends up doing is generating this double bond to the nitrogen. And that then can release the ammonia through an attack with water, and the product of that then is alpha ketoglutarate. Yes?
Do you mean NADP or NAD?
Yeah, NADP plus or NAD plus or NADP plus. Okay. In other words, here what it's saying is either NAD plus or NADP plus can pick up the electrons and come off as NADH or NADPH. Well, I see what you're saying. Okay. Another error. Okay. Dum-dum-dum-dum. All right. I will get that as well. Since this reaction is a freely reversible reaction, if there are circumstances where you're trying to fix ammonia, it is also one way that that nitrogen can be fixed. Now, we've seen in many contexts along the way that when we want to do something biosynthetic, we have to activate that thing somehow by adding phosphates to it, by adding sugars to it, whatever. What we're going to do next is we're going to activate that nitrogen in a way that allows us to manipulate it further. Sir, question? And the first step in doing that is we're going to make a carbonyl phosphate that ATP can pick up a bicarbonate and generate a compound called carbonyl phosphate. So this is basically a phosphorylated bicarbonate, if you want to think of it that way. And you can imagine that that would be a fairly reactive compound. And the ammonia can attack then and produce a compound called carbamate, looking like that. Second molecule of ATP used, and now we have carbamyl phosphate. So carbamyl phosphate is going to be the activated form of the ammonia that we need in order to make a urea. And we're going to dispose of that nitrogen through another metabolic cycle. We've seen the power of cyclic metabolic pathways where you can basically feed something in, get something complicated to happen, and pull out what you want. And that's what the urea cycle is going to do for us. Here at the top we see the carbamyl phosphate, and it's going to be plugged onto the end of an ornithine to make a citrulline. We're going to take in an aspartate and sort of smoosh that onto the other side of the carbon, and then be able to remove just a carbon dicarboxylic acid. We now have made an arginine. So we've made an arginine, which we're then going to use to make urea. We're going to split off urea, and that regenerates the ornithine. This is the overview of it. We're going to be picking up at least one nitrogen from here. We're going to be picking up nitrogens that from the ornithine and coming off as the urea. So let's take a look at how that happens in detail. Here is ornithine. It probably got its name because it's abundant in bird poop. And it is a happy recipient of the carbamyl phosphate, which can then add on to that nitrogen, losing the inorganic phosphate. So that's what drives that reaction forward. And now we've got citrulline. Citrulline is moderately abundant in watermelons in its name. That enzyme is called ornithine transcarbamylase because it's transferring a carbamyl group. The citrulline we've just made is then going to be a target for having an aspartate added to it, which seems kind of like a large, bulky thing to add. And the thing to look at in this case is this nitrogen, which is going to replace the oxygen over there to make argininosuccinate. That cost us 2 high energy bonds to do that. This argininosuccinate then can be split right there. And as you might guess, that's basically a free reaction. And what happens is, in splitting off a molecule of fumarate, you release a molecule of arginine. And if you look in here, you can see that we've got the components of this almost to be a urea molecule from 3 different places. There was one from the ornithine, one from the carbamyl phosphate, these from the carbamyl phosphate, and this one from the aspartate. Arginase can split right there by adding some water across it, and that releases urea as a molecule. And we've regenerated now our ornithine, which looks a little bit like lysine at a different length. This intervention here, the fact that ornithine is part of the urea cycle, is the reason why arginine is and isn't kind of sort of an essential amino acid. Our body's ability to make enough of it depends on how heavy the demand is and how rich the diet is in it. You've noticed, I think, already that there's potential for connecting the urea cycle to citric acid cycle intermediates. And these interactions are sometimes called Krebs bicycle. So that the aspartate that comes in to the urea cycle to make your argininosuccinate and carbon backbone comes out as fumarate, malate, oxaloacetate, and then that gets transaminated to make aspartate again. So this step is going to get rid of a nitrogen by using this citric acid cycle intermediate. And then up in here, of course, we got rid of another nitrogen in order to make the nitrogen we put into the carbamyl phosphate. So 2 nitrogens go in, 1 urea comes out. There is some regulation of the urea cycle, as you might expect. The urea cycle is a fairly energy-intensive cycle. You noticed a number of places where it's going to consume nucleotides. It's also dealing with a precious resource. Nitrogen is never to be dismissed lightly. And so the urea cycle is regulated. If, for whatever metabolic reasons, you are engaged in high levels of breaking down amino acids, if their catabolism is high, then the concentration of glutamate is going to be high also. That will tend to increase the synthesis of something called N-acetylglutamate. And N-acetylglutamate is an allosteric regulator of the carbamyl phosphate synthetase. So carbamyl phosphate is the committed intermediate going into the urea cycle. Now, what's interesting, in order to make N-acetylglutamate, we also consume a molecule of acetyl-CoA, which is another way of tying its regulation to the energy status of the organism, that there needs to be a certain amount of acetyl-CoA around from fatty acid breakdown if you're going to want to make glucose out of the amino acids. Now, urea is a solution to a specific problem, the fact that you and I and most mammals have a reasonable amount of water available to us. Birds are very much concerned in trying to stay light. Reptiles often live in dry climates. And so both birds and reptiles have a slightly different mechanism for disposing of excess nitrogen. They start from xanthine, so it's a purine ring, and a xanthine oxidase, which consumes oxygen, makes hydrogen peroxide of all things. And now we have uric acid, and uric acid is not very soluble. It precipitates nicely. So if you think of some of those trees near the old Capitol where you don't want to park your car, there's a fair bit of uric acid there. There's also a certain amount of uric acid produced from the breakdown of nucleotides. So you and I also make some uric acid, and it is precipitates of uric acid that are the culprit in gout or in some kidney stones. So although we don't use this as our principal pathway, it can cause trouble for us. There are another couple of strategies involved. So organisms that have access to urea and want to recover the nitrogen out of that can do so using an enzyme called urease. And urease can simply use water to break that down and recover the ammonia. Urease was the first protein to be crystallized to prove that it's a single thing. And the ammonia released in that way can then be fixed for future use. Fish, by contrast, are completely surrounded with liquid, and they simply, wash away their ammonia and don't worry about it further. And I think that's the end. Have a good weekend.